Hello everyone. In the previous lectures we have talked about evolution. First, how did we get to know that organisms actually do evolve, they're not produced as is. Second thing, we talked about microevolution, so those genetic changes that take place within populations. And then we talked about macroevolutions, so those larger changes that have ultimately result in the formation also of new species. What we're going to be talking instead in this lecture is about systematics, and how systematics basically has the double function of reconstruct all of these evolutionary events, and by doing that is basically trying to produce two things. It's trying to produce, first of all, the relationship between the different organisms in terms of from an evolutionary point of view. That's the point of phylogeny. And also then classified all organisms, again reflecting not only the evolutionary relationship but also their morphology. So these are the topics we're going to talk about here. First, we'll define what systematic is. Then we'll talk about how the still currently used classification is, so which is the one based on Linus' discovery and or method. And then we'll talk about characters. So you'll see that organisms are, or the evolutionary relationship between organisms depend on similarities and differences, ultimately on characters, on phenotypes that do present. We'll see what type of phenotypes or characters are studied and how they relate to the type of evolution of different organisms and what are the problems when you use them as well. Then we'll talk about phylogenetic trees and how they basically portray in a graphic way all the finding in terms of evolutionary relationships between organisms. And then we'll finish up talking about first cladistics, this new field. Well, new field, it's been around for 67 years, but a field that basically tried to put a different point of view to systematic and phylogeny. And then we'll finish up talking about how molecular data are helping nowadays to further refine the relationship between different organisms from a systematic and phylogenetic point of view. So let's start talking about systematic first. What is systematic? Systematic is that field that studies how the relationship between organisms in terms of evolutionary relationships. Okay, and so it has two main goals. The first one, again, is to reconstruct the relationship, so reconstruct the phylogeny or evolutionary history of these different organisms starting from the original common ancestor and how close they're related and so on. And then the second goal is to actually place them into groups. That's what taxonomy does. Place them into groups so that they can be classified and named so that the international community can use a common language ultimately to refer to specific type of living organisms. Now this leads to what is called classification, where all the organisms are put in hierarchical group. Now the system that has been used since the 1700s and is still used nowadays, even though cladistics is planning to change it, I will talk about that later on, is the system that was proposed by Carolus Linnaeus, which basically was placing all different organisms based on morphological similarity and later on also evolutionary relationship into fixed groups, categories referred to as taxa. So each of these categories here referred to as a taxon, which in plural is taxa. So now these are all hierarchical groups. What it means is that all organisms will first start all of them being classified as a large group, and then they slowly will be divided into smaller and smaller and smaller taxa, where you have less and less and less organism until you reach the species itself. So obviously all life first is divided into three main domains, and then from there you would have more kingdoms, so less organism in a kingdom, and you would have phylum or phyla plural, classes, orders, family, genus, and then you finish up with the specific name of the species, so based on all hierarchical taxa. Here is an example of how it works. For example, for this beautiful bird, which is the cardinal. Obviously, it's a living organism, so it belongs to life. And then it belongs to the specific domain called eukarya. Okay, it's an eukaryote. Among them, there are plants and so on, fungi and so on. It belongs to Animalia, so the kingdom of Animalia. Then it belongs to a smaller group. There are the vertebrates and invertebrates, so it's a chordate, and then it's a bird, and so on. As you can see, the linear classification, again, places this bird starting from larger groups corresponding to domains into smaller group until you get to the genus and the specific species you're talking about. So by using these words here, now scientists can quickly identify any species. Over the phone you can say, oh, I'm talking about Cardinalis. You would know exactly what type of organism you're talking about because linear classification has been used to place it. Sorry, there was a mistake on top. I had a different example, Canis lupus, which is the wolf. Now instead I change it to the bird, to cardinal here. Now, just to remind you, I already mentioned this when we talked about macroevolution. Species are named nowadays using a binomial nomenclature, which consists of two parts. The first part corresponding to the genus, which in reality includes several species, and the second one, which is the specific epithet, is the one that really defines the specific species. Okay, so for example, for bears you have the Horses maritimus, polar bear, and the Horses arctos, brown bear. Now sometimes you may even see a third name there, and that's what is used when you want to specify a specific subspecies. For example, Homo sapiens sapiens is a subspecies of Homo sapiens. Now let's move on. Let's talk about what the characters that are used by systematic in order to recreate the phylogenetic relationship between organism and classify them. So we're talking about the systemic characters and how they relate to the type of evolution. So these are referred to as systematic characters. So now these might be referring to character that give similarity or differences between different organisms. Okay. Now one important thing, systematic and phylogeny are based on evolution. And evolution, I hope you got from at this point from the previous lectures, as you know, acts on genetic material. Okay, so systematic, since it's trying to represent, particularly with the phylogenetic trees, the relationship, the evolutionary relationship between organism, can only focus on genetic characters, not environmental character. So here, for example, you have an example that I already used when we were talking about variability among different organisms, and this was used as an example of environmental variability. This is indeed the same plant, which can have flowers of different colors depending on the acidity of the soil. But you can see how if you're a systematist and you're trying to study a plant and you're focused on a character that doesn't have genetic evidence, you may actually be misled. So in this case, it will make you think that you're talking about two different species, but no, it's the same species, just in different environments. Similar, this example here, if you look at this, wow, these will look like a beautiful flowers. So if you start spending time studying this, you're actually misled, because this is indeed another example of an environmental type of characters. This is indeed a flower-like structure. It's not a real flower, but a flower-like structure which is formed by a parasite of this plant, which is the parasite is the fungus Puccinia monoica, which actually causes overgrowth of the leaves and change of the color to simulate the presence of a flower, so that now actually this also leads to production of different scents. So now that you may have pollinating insects being attracted to this part of this plant and spread the spore of the fungus. So this structure, again, it's environmental. In this case, it's caused by, is not related to the genetic of the plant, but to the infection by a fungus. And therefore, this is not one of the characters that should be used in systematic characters. Now, so systematic characters focus on genetic characters to figure out the relationship between organisms, not the environmental one. Now, even when it focuses on genetic characters, now it focuses on genetic characters that should be independent from each other, genetically independent from each other. What do we mean by that? Well, look at this picture here. So now you may have that different characters in an individual are actually the product of the same gene, the same locus or the same gene. It's not the same thing. I remind you the locus is just a sequence of DNA which is present always in the same location and conserved in a chromosome. Might be coding for a protein. That's what systemic characters or classic systematic is using, because that production of a protein will result in a phenotype, or it could be just a sequence that is conserved, not necessarily producing a protein. So going back to our example here, so you may have the different phenotypes are produced by the same coding gene. Right? There is no point of studying all these characters for a systematist, because obviously since this gene is the same, now you will find the exact same relationship between individuals carrying this gene in an evolutionary tree. If they have character A of a certain subtype 1, even character B will be of the same, related to that one. It will be, this character will always be connected with each other. So what a systematist wants to focus is on as many as possible characters that are caused by different genes, because all of these genes are subject by different pressure from evolution and might change differently in different organisms. Now, these characters, depending on what is the mechanism of evolution that leads to them, can be referred to as homologous or analogous. We'll see a second. So what are the different type of evolution, first of all? So the point is that if you're taking different organisms and now you're looking at their similarities and differences, so you're working as a systematist now, and you see, well, they actually both have wings. They have different, but one of them has two, let's say 6 legs. The other one has only 4. Are they related or no? Well, these similarities and differences do not necessarily always allow you to make the jump to a closeness between these organisms. The reason being that they may be produced by different type of evolution. So there are 3 main types of evolution: what we call convergent evolution, parallel evolution, and divergent evolution, which can affect characters in a different way. Let's go through these examples. So first of all, convergent evolution. So we mean by convergent evolution that type of evolution that take place in 2 different organisms which are not related by a recent common ancestor, okay, but that ultimately leads to them to have a similar character. So this type of character, first of all, will be referred to as an analogous character, and again is produced in reality by 2 evolutionary events. It has evolved twice. It's not that it was present in a common ancestor and then was obviously inherited by both type of organism. No, these 2 organisms are independent from each other from an evolutionary point of view, at least recently, and then independently they have evolved exactly the same character, the same analogous character. Here you have an example of what happened in the cacti in North America. So that's the cactus, and then you have the spurges in Africa. They look extremely similar if you look at them. But if you actually look at their most recent common ancestor, this character here that you're looking at that would make you think that they are closely related was not present. So meaning that this character has evolved twice, once in the cacti in North America and once in the spurges in Africa. So 2 events which will mislead you to think that these plants are closely related, but instead they are just the product of what is called convergent evolution. Another example is the wings. If you look at the wings, there are at least 4 different types evolution that took place there that led to the formation of wings, which corresponds to the animals that have it. You can find them in birds, where you have that the wings are actually made by the obviously there is a skeletal part here, and then there are feathers. If you look at bats and pterosaurs, they're made by an extension ultimately of the skin there, and then you have in insects that is actually made in a very completely different way. There are not even bones there inside there. So the fact, if you look at flying, you might think, well, this is possible that these four organisms are related to each other, but no, in reality they're very, very distant from an evolutionary point of view, right? So these are reptiles, these are birds, these are mammals, these are insects, very different. We know that now, right? And indeed, this is just the product of convergent evolution. So these structures to fly have evolved actually at least 4 times independently from each other. Again, this is a type of analogous type of character. Now, if you look only at the pterosaurs, birds, and bats, you may actually say that, well, it's true. If you look at the surface that it's used to fly, yes, this is an analogous type of character. But now, if you look instead at the bone structure, which instead has evolved through a common ancestor, so it's first forming a common ancestor here. They're all tetrapods. Now, if you're referring only to the skeletal muscle of this, actually this is the product not of convergent evolution, but divergent evolution, and this is referred to as a homologous type of character. We'll talk about that in a second. So, convergent evolution, you have two organisms. They are not closely related to each other. They don't share a recent common ancestor, but that by chance they have developed, or because of the common pressure they're subjected to, or similar pressure they're subjected to, they have developed analogous characters, so similar characters between each other. Now, this may happen also by parallel evolution. In this case, now the organism may have actually a common ancestor, but still their similarity does not reflect the presence of that character in the original common ancestor, and is still the results of two evolutionary events. You have an example here, the flying ability of what is called the sugar glider. So this is a mammal that lives, or marsupials mammal, that lives in Australia. And then here you have the flying squirrel, which is a placental mammal, fairly different from marsupial ones, that lives instead in North America. They all share a fairly common recent ancestor, which is a rat-like type of animal. Not surprising, if you look at squirrels, they look fairly similar, right? And however, if you look, this common ancestor did not have the ability to fly. Did not have the skin and hair flaps here, joining the forelimbs with limbs on the back. So this character here is the product of two evolutionary events that happen in a parallel type of evolution in this case. The third type of evolution that can take place is referred to as divergent evolution. So in this case, what you have is that the difference, we're not talking about similarities in this case, we're talking about differences, and the character you're looking at is referred to as a homologous character, not analogous, but homologous characters, are nothing else than the product of divergent changes starting from a common ancestor that had the same structure. So an example is what happened here to the limbs or of different animals. So this is what will be our arm. Now it turned into a leg in horses and cats. In bats, has turned into the structure that gives origin to the wings. Same thing for birds, and for whales actually has evolved into their flippers. So these structures, they all evolved through divergent evolution, so they're all homologous structure. They've evolved from a common ancestor here, was a tetrapod, which then they have adapted to different environments, as basically acquiring differences, okay, among them. So this is a type of divergent evolution, and this type of character you're studying here will be referred to as homologous character. Another example of homologous character, very interesting one, is the one related to the bones that you find in your own ears, for example, or in different vertebrates, including also reptiles, mammals, and so on. So I'm not sure if you are familiar with that, but if you look at the way you perceive sound, for example, you know that your sound is captured by your ear, and then it travels through the air, through the ear canal, and then it gets transferred from this air environment to an aqueous environment inside your ear, which correspond to this structure here, which is the inner ear. This is referred to as the cochlea. Now, this is filled with a liquid. So the vibration perceived by your ear have to be transferred to the liquid inside here. And this is done through the tympanic membrane. There is a membrane here, or eardrum, or tympanic membrane here, which then, through 3 bones, transfer mechanically the vibration of this membrane to the aqueous environment here, to the inner ear here. And then the movement of the water is perceived by an interesting type of epithelium, which has cilia that moves and so on at different frequencies. Beside that, so now, if you look at these 3 bones here, they are actually evolutionarily coming from the skull of the early jaw fishes. They're actually bones that were present in the jaw of very, 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 very old type of fishes. Okay, and then they have evolved both in reptiles and mammals, forming this. And they have first evolved also to support the gills. You can actually see that from an embryological point of view. If you look at a human embryo at 5 weeks, you can actually see these bones here. They first resemble, they give a structure that resembles indeed gills, and then they get assembled together in the middle ear here, allowing for the transfer of the sound. So again, these are homologous characters, what you're looking at here. The structure of these bones are homologous structure to the one that were present in the fishes, and they have diversified throughout evolution to the point that they became a structure for perception of sound in vertebrates and in reptiles and mammals through divergent evolution. Now, this is based, what we talked about is all characters. They're visible characters that we refer to as overall as morphology. Now, systematics and phylogeny uses other sources of information, such as the anatomy of the chromosomes. If you remember, you can do the banding, look at the structure of the chromosome and see the differences, the deletion, translocation, and so on, to get more information about how close pieces are to each other. The structure of subcellular structures, they look at different organs, organism behavior of animals. It's another factor that is used, and we'll see later on how molecular data, so nucleic acid sequences and protein sequences, are also used now to further define the relationship between different organisms. Now, all organisms ultimately will result in the combination of early type of characters, what is referred to as ancestral type of characters, and new characters that have been derived from the original ancestral character derived characters. Okay, so there are mosaic, if you look, all of these type of characters. Now, so the character, the original character, for example, in this case here, when we're talking about these jaw bones, right, this will be the ancestral character, now has become the derived character, which will be this one here, the bone in the ear, in mammals, for example. So the same character can be defined as ancestral, how it was before evolution took place that created the derived character. Okay. Now, the how do we know what was coming first and what was coming last? How do we know, for example, that it was not the other way around, that these bones were, obviously, it's a ridiculous example. Now we know how much more recent humans are, right? Obviously, you have a lot of background information already when you're watching this lecture, but imagine you're one of the first systematists and still don't know about that well about evolution and so on, the timing of evolution. Don't even know how old the Earth is. And now you see, were human first and then these gave origin to fishes and their jawbones and so on, or vice versa? Which one is the ancestral and which one is the derived character? How do you figure that out? Well, there are 2 different ways you can go, or 2 main ways. There are different other ways you can go, but in general, you can either look at fossils that predate the organism you are taking into account, or you can look at an outgroup, so some unrelated organism that somehow that can be related to this and possibly older organism than the one you're looking at. So one example, for example, talking about the vertebral column. Now there are organisms, vertebrates, that have it, and organisms that do not have it, invertebrates, such as jellyfish, whatever, or insects, and so on. Which one came first, right? How do we know that invertebrates actually came first, and therefore the lack of vertebral column was the ancestral character, and the presence is the derived character? How do we know about that? Well, you can do that by looking at old fossils that do predate vertebrates. If you look at very old fossil, you see that none of them would have the vertebral column, therefore pointing to the fact that early organisms were invertebrates, did not have it, so the ancestral character was the absence of vertebrae and the vertebral column, and the derived character is the presence of vertebrae and the vertebral column. Another way to do it is by looking at what we call an outgroup comparison. For example, you're now looking at two type of organisms that are very closely related to each other, like these butterflies. Okay, there are two types of butterflies. They're very closely related from an evolutionary point of view, but some of them have 6 legs, and some other ones still in reality have 6 legs, I must say. But it looks like they have 4 of them. So they're defined as 4-legged butterflies. This is the monarch. You see it all the time in Toronto. In reality, they have 6 of them, as I was saying, because the anterior ones, so the one closer to the head, are still there. The only difference is that they're short and curled towards the body, so you don't really see them. All right. But now, what is the ancestral and the derived character in this situation? How can you figure out this type of butterfly come first or after? So is the 4-legged the derived character or the ancestral character, or vice versa? How do we know that? That this indeed is the ancestral character. The 6 is because if you actually look at an outgroup, in our case you just have to look at insects. They're both insects. There are insects that are much older than these organisms. Just look at them. You see that insect by definition have 6 legs. So pointing to the fact that this must have been the older ancestral character, and this is the derived character. So let's move on. We talked about all these characters here. Now, how can they be used to summarize the evolutionary relationship between different organisms? And this can be portrayed using what we call phylogenetic trees. So these are just schematic diagrams that all they do, they just summarize the finding in terms of study of the different characters. So they summarize them in a visual way, which tells you how close species are to each other. So basically, phylogenetic trees, what they consist of, and as the name says, they're nothing else as trees. This is an original one done by actually Darwin, one of those that he was graphs that he did in his own notes, trying to figure out the relationship between different organisms. And what he was saying is basically that all organisms, as in any phylogenetic trees, they are originally joined together, and then with time they just separate evolutionarily, right? And so each node represents what is called a common ancestors, and then each line represents the new species that will come out, and then the same species can further split. So if you think, this is nothing else than the graphical representation of macroevolution taking place, giving origin to all the different species. Now, it gives not only information about the relationship between species. For example, you know that these two species in these two branches here will be closely related or closely related compared to this one right here, but it gives also information in terms of time, right? The farther you are from the common ancestor, the more distant you are in terms of time overall. So as you can see here, for example, here, distance between gram-positives and planktomyces, I wouldn't even know what they are. You can see they're fairly close. The distance here tells me that they're overall fairly close from an evolutionary point of view. Much more closer than they would be compared to our aquifex here, okay, in terms of time. So it gives information about the relationship between the different species and the timing in which these different species have form and their distance from other species. Now, these branches, they get smaller and smaller over time, like in a tree, perfect tree. So the larger you are at the beginning, the more species will have been included there that just then they have come out over time. So now, how are they generated? Well, an important factor that is always used generating the phylogenetic tree and reflects the fact on how evolution actually works. Evolution tendentially try obviously to get organisms to adapt to a specific environment, for example, adapt to swimming or adapt to flying and so on. But it doesn't keep doing it all the time. Okay, once it gives an organism a specific structure to adapt, it tends to keep it. All right, and these events are rare overall, right? So meaning that the formation, for example, of fins, it will happen once, twice, but it doesn't keep happening all the time, right? And so, and that's the reason why when you make phylogenetic trees or hypotheses, you use what is called the assumption of parsimony. What you assume is that any new character that is you find most likely is due to the smallest number of possible evolutionary events. So if you find fins in many animals, right, you're not start thinking that for every animal has evolved again. What you're gonna do when you create a phylogenetic tree, you try to put them all together, possibly. Okay, unless there are other evidence that suggests that they may not be put together. Okay, by putting them all close together in a phylogenetic tree, you're saying, well, that event, the formation of the fins in this example, has happened only once. The same for the wings, the feather wings. They evolve only once, not multiple times in each bird type, right? So you use what is called the assumption of parsimony, being parsimonious, being cheap, right? You don't want to repeat the same thing over and over. You make the assumption that everything that is new most likely has been caused by one or very few evolutionary events. Now, the systematists, they use these phylogenetic trees ultimately also to place organisms, to classify the organism and place them in what we call taxa. As we say before, taxa correspond to those groups such as domain, kingdom, phylum, genus, families, and so on. Now, these taxa, they don't necessarily always reflect, and I'll show you an example later on, they don't necessarily always reflect a group of all the groups of individuals that share a common ancestor. So now, there are different types of taxa. You can find 3 of them, okay? And that you can find in systematic as well. So what we have is monophyletic taxa are basically groups that are formed by all individuals that share the same common ancestor. So look at this. You have these 2 species and the common ancestor all grouped together in the same thing. That's a monophyletic taxa. This is also referred to as a clade. You see when we talk about cladistic. Polyphyletic taxa instead are groups, could be a kingdom, a phylum, that because of morphology as well, puts together organisms that don't necessarily share the same evolutionary lineage. These are different than paraphyletic taxa, which are overall similar to monophyletic taxa, but still exclude some of the organisms that have the same shared common ancestor. For example, if you look at reptilia and birds and so on, are referred to as paraphyletic type of taxa. Let's show you the example here. What do we mean by paraphyletic? So if reptilia here, so this is a class, okay, that includes all organisms with specific evolutionary history, not only that, but specific also morphology, okay. Now, if you look at reptile and you look at phylogenetic tree, you see that reptiles includes a common ancestor here, different type of organisms such as turtles, lizard snakes, crocodiles, but then do not include another group that evolutionary is related to taxa, which are actually birds. So they don't include all the organisms that have originated by the same common ancestor, but only some of them, and therefore is referred to as a paraphyletic taxon. If it was a monophyletic type of taxon, it would be expanded to include also birds. Okay, and that will be also referred to as a clade. Now, summarizing what we said until now. So we talked about taxonomy and phylogeny. Okay, so how all of this is done, first of all, by studying different morphological characters, but now there is also molecular data, behavioral factors, and so that feed into them. Now we talked about convergent and divergent type of evolution, and also we talked about parallel type of evolution, and how the characters are defined differently as homologous or analogous depending on whether they are from convergent and divergent type of evolution, and how once you put them all together using the principle of parsimony, they can be used ultimately to recreate the phylogeny, so the relationship represented by phylogenetic trees between all different organisms, right? And then this phylogeny is used by taxonomy to try to group all different organisms into different convergent groups called taxa, hierarchical and convergent groups, as the linear taxonomy uses, right? And this is done by using different type of taxa that can be monophyletic, polyphyletic, or paraphyletic. Okay. So now, so as we said before, the systematic or linear systematic now, or I just mentioned in the previous slide, uses not only the evolutionary information that you have about living organism, but also other morphological characters in order to classify an organism. And one example is on how birds and reptiles are actually grouped into different classes, because of, even though they have the same evolutionary origin, which would theoretically should make them put all in the same group, now they have some morphological properties like the presence of feathers that makes them different, and therefore they're classified into different groups there. However, as you can see, there is a bit of a problem there, and you can see it here. So for example, this is how the saltwater crocodile called Crocodylus porosus is classified. So it's an animal, it's a vertebrate, it's a reptile, and then you get into the order here. Now, this is how a bird, such as this beautiful blue jay, or Canada here, let's sing it, is classified. Evolutionarily speaking, they basically should be extremely close, and you would expect that the taxonomy should reflect that, but it doesn't. Look at the classification here. The only common thing that they have, these two type of animals, are the kingdom and the phylum. After that, you have the class, order, family, genus, and species that are completely different here. So there is some discrepancy between what evolution says and what the classic taxonomy is saying here. It's placing organisms that theoretically should be fairly close, evolutionarily speaking, far away. All right, and that's exactly what cladistics is trying to address. Okay, cladistic is basically a subfield of phylogeny and systematics, okay, which has emerged about 60, 70 years ago because of the work of the scientists, you don't need to remember. And now, what it's trying to do is basically suggesting that organisms, instead of being grouped, okay, and the same thing, phylogenetic trees, instead of taking into account not only the evolution but also other morphological characters, so both systematic and phylogeny should forget about the ancestor character and focus only on the derived characters. So basically focus only on evolution. What it's saying is that represent animals, group them together just based on evolution. So just don't keep looking at the ancestral character that may be still carried over or other morphological information. Group animals by just by derived characters, just following evolution. And basically what it's suggesting is that organism should always be put in what we call clades, so in taxa which are monophyletic, only monophyletic, and they reflect the common ancestor and all the organisms that have originated from that point on. So just to give you an idea. And this is represented by what they generate, which is cladograms. Cladograms are nothing else than phylogenetic trees, okay? But they do follow the cladistic rule, where all they follow is the evolution of organisms, okay? Not necessarily other morphological data present in them. And so basically what it does is start from all organisms and it slowly divides them into monophyletic groups or clades based on one specific derived character that you are taking into analysis, referred to also as a synapomorphism or synapomorphy. If it's one, synapomorphism is the plural of that, and which basically causes these organisms to branch in a subclade. But it's always generating basically clades. So look at this example. So there will be more than 3 clades, but here this will be one clade, one monophyletic group. This is another clade, so it has the common ancestor and all the species generated from there. This is another clade. Now the common ancestor is here, and these are all the organisms that have originated from that common ancestor. In the way these groups of animal are split progressively one after the other, again, is based on a specific derived character. Okay. For example, this one here will be the presence, of the placenta, or and so on. The presence of feathers here, other characters that you can take into account. That's not a point of this slide right now. So by doing that, again, it's not only changing the way phylogenetic trees should be produced, and they shouldn't be too far away anyways, too different than the classic phylogenetic trees, but also it's suggesting that now organisms should be also named following different rules than the one followed by the linear classification. Again, linear classification, I repeat it over and over, is based on evolutionary information and different morphological characters. Okay, you combine them together, but their final results does not necessarily always reflect pure evolution. Phylocode, which is basically the new international code of phylogenetic nomenclature, which is suggested by cladistic, instead suggests that you should just follow evolution and group, instead of using taxa based on both information, evolutionary and morphological, should just produce this taxa just based on evolution. So just following the clades. Just to give you an example how it changed or how they suggest to change, keep in mind this classification is not the official one. It's just to give you an idea of how it would change. Now, so this is the classification, as I said before, of the saltwater crocodile and the blue jay based on the classic linear classification. So you have the kingdom, phylum, and so on, Animalia chordata, and so on, which does not necessarily reflect their evolutionary relationship, because it's creating reptile, as we said before, as a paraphyletic group. So including only some, the common ancestor, but only some of the animals originated from that, and excluding birds. What cladistic instead is suggesting is, instead of doing this artificial separation based on just some additional morphological information, you can just group animals and classify them based on the clades they belong to in every evolutionary step. So if you look at this point, and again, are not the official, okay, there's a lot of rules that are used by Phylocode to classify animals. I'm not following them here, it's just to give an idea. But now you see how the saltwater crocodile now will belong first to this clade, so Tetrapoda, and then it will belong to this clade here, Amniota, Sauropsida, and so on, and progressively will belong to smaller and smaller monophyletic taxa or clades. But you can see how ultimately now this classification will reflect better, if you want, the relationship or evolutionary relationship between these two animals, which morphologically they may look very different, but again, evolutionarily are very close to each other. Say before, cladograms are nothing else than phylogenetic trees that are based only on evolution. Okay, so derived characters, so how and which are driven by evolution, so are only represent evolution, and they are originated by looking at different specific characters, okay, which can be morphological characters and how they've changed over time, and they're used that to basically progressively separate a specific group of organisms. For example, in this case, you could generate, and we will do that, okay, in a live meeting. I'll do that together with you in a live meeting. So you can actually separate all these group of organisms that you have under your analysis right now based on different derived characters, which is the presence or absence of vertebrae, the jaws, and so on, the swimming bladder, feathers, and so on, into smaller and smaller clade, coming out with cladograms that look like this. So for each divergence that you see here, this divergent evolution here taking place, you would have a specific synapomorphy, as we said before. In this case, the presence of vertebrae that would split, for example, Lancelet here, which doesn't have vertebra, from the other organism. So at this point, this branch here would include all the other organisms there. And from there on, now you will look at a different derived character. In this case, will be the presence of jaw, which will separate at this point the lampreys that don't have it from all the other organisms. This is the other derived character we are looking. And so you can follow that and slowly, basically, branching out different organisms, creating your cladogram, and basically building smaller and smaller clades, which then could be used also to classify these organisms using the phylocode. Last part of this lecture is talking about molecular data. As I mentioned before, now we have what we discussed until now is based on morphology ultimately. How morphology has to be obviously related to some genetics. Okay, so it has to reflect genetic characters and so on, and ultimately should reflect the type of evolution taking place. Nowadays, in the last few decades though, we have accumulated more and more and more data in terms of genomic and mitochondrial DNA sequences, RNA, protein sequences, and so on, which now can actually, and we get them from all sorts of organisms, so many organisms, and sometimes even some DNA was found also from fossils too. So now all of these data can actually be used nowadays, instead of using morphological characters, can actually be used to further define the relationship between different organisms. And so basically to see how close they are related to each other and how long ago they have separated. Now, using molecular data compared to morphological data is that you're actually studying the first, represent different advantages and disadvantages, right? The first advantage is that we have so much data right now. Okay, so we have so many type of genomes. Not only that, but now instead of relying on how this genome produce some phenotypes, some characters, you can go straight to the source. At this point, you can actually study not only phenotypical variation, but even variation just in sequences. As I said before, loci are not always coding for proteins. So if you look at morphological characters, you're just looking at the variation related to coding genes or coding loci. You can actually, there are loci that don't code for proteins. They're still preserved throughout evolution, and they change over time. You can even use that. So you can have so much data now to further define the distance or the relationship between different organisms. Now, the other advantage is many genes are conserved throughout evolutions. And so you can really, say if you take the same gene or same sequence, even if it's not a gene, you can just compare it. You can use small sequence or large sequence if you want to divide even better many organisms. Now, there is obviously no effect of the environment. Well, there is effect, but it's not going to be carried over for many generations. So whatever difference you see, you're guaranteed it's genetic and not environmental. And now the other advantage I'll discuss in the next lecture is that it also allows you to further define the distance in terms of time between organisms. When you look at morphology, yeah, you can say, okay, these two organisms can be close to each other, they have evolved. The same derived character has evolved in two different locations, in different directions, right, by divergence. But how long ago have they separated? You got a date, fossils, and so on. In this case, if you look at the DNA, the amount of difference that you see in difference, it usually relates to the time in which these two organisms have split. There are always some drawbacks as well as everything, but in the end, the advantage greatly outweigh the drawbacks in this case. So first of all, there is no always a relationship between a change in function, which may mean that some changes that you see are really meaningless. Particularly, consider the fact that some changes, I mean, can be just random. If you think you have only 4 nucleotides, so if you're looking at a nucleotide in one position, and you see some organism, for example, have an adenine, other they have a thymine in that location, it's just 1 out of 4. How do you know it's really meaningful, right? So you have to obviously analyze larger sequences to really figure out relationship. Same thing with 20 amino acids. It's already better, obviously. Statistically, the chance of having the same amino acid in one location is most likely due to preservation or acquired derived character that is kept there more than just by random chance. But again, still 20 amino acid, there is still a chance that you might have convergent evolution instead of common divergent evolution taking place. So as I say, one big advantage of using molecular data is that it allows you to actually also measure not only the relationship between different organisms based on the percentage similarities between the molecular data, but also their distance in terms of time. Okay, and this because mutations, for example, in the DNA or changes of in amino acid in proteins, ultimately they take place at a specific rate. This is not always the same. Okay, it depends on the type of molecule that you're using. Okay, and the type, for example, even between proteins, it depends if a protein is essential. Say, okay, it's a protein, for example, part of the in the chloroplast of the photosystem. Obviously, it's an essential protein. There is very little selective pressure towards it. It would tend to be kept. For example, Rubisco. Rubisco is the gene, if you recall, the first gene in the Calvin cycle. It's been preserved almost as is for billions of years, right? The reason being that there is no selective pressure on that one. It's so essential that it cannot really change much. So it depends on what you're looking at. But for certain type of categories, like mitochondrial DNA, for example, mutates very quickly at a constant rate. If you look at ribosomal RNA, is lower, but still at a constant rate overall. So by figuring out what is the rate that you get there, you can actually figure out also the distance between organisms. Here is an example. If you look at the beta chain of the hemoglobin, it's conserved among all these different organisms. So gorilla, monkeys, mouse, chicken, and lampreys here. If you look, this is made in human by 146 amino acid. If you look at the difference in terms of amino acid, you see that the difference there indeed relates to the evolutionary distance that there is between humans and these other organisms. We're very closely related to gorilla. You should see me in the morning to know that. So, and indeed, the difference, basically beta chain is the same. There is only 1 amino acid difference. We're a bit more distant to monkeys, and so we have 8 amino acid difference there. We are so distant from lampreys that basically you're talking about a completely different protein at that point. You have 125 amino acid different out of 146 between humans and lampreys. But again, this just to show you how indeed the distance, evolutionary distance, relates to the differences between, in this case, proteins, which then could help you to figure out the timing of evolution or separation between species. Now, when it comes to mitochondrial DNA, it's very commonly used, okay, and it's one of the type of DNA that evolves fairly quickly. It's non-essential, it doesn't code for that many genes, okay, and therefore it tends to mutate faster than other type of, for example, compared to chloroplast DNA, which instead codes for more genes that are essential for photosynthesis. So it evolves more quickly, and therefore, because of this faster evolution, can be used also to study recent evolutionary divergence. Let's say you want to study, and we'll do an example. We'll go through that. You want to study how far humans and gorillas have separated. How long ago? You can use mitochondrial DNA. We're fairly close to each other in terms of few million years distant. Same thing with chimps. You can use mitochondrial DNA. However, it's a problem if you want to use or try to distinguish or figure out how long ago we have separated from lampreys, because we are so far away that basically the mitochondrial DNA, first of all, it will be basically completely different. You have so much differences that at that point becomes less helpful even to define that distance. So it works well in short distances, evolutionary distances, but not that well in large one. Instead, molecular data from like chloroplast DNA, ribosomal RNA, or highly conserved proteins, so structures that don't change that fast, is that are much more helpful to define relationship between organisms that are much more distant from an evolutionary point of view. We're talking about hundreds of millions of years at this point. So when it comes to analyzing DNA, and we'll do that, I'll guide you through an example using one of the many tools that you can use to that. In our case, we'll do an experiment using primate DNA. We can see how we can use mitochondrial DNA to actually recreate the phylogenetic tree of hominidae. And so how is the analysis done? Well, the analysis is done by basically taking the sequences of DNA, and first of all you align them with each other. The first alignment already shows you differences, right? But also shows you potential deletions, translocation, insertion, and so on. And these are already information that can be important. If you see that they are conserved among different organisms, even insertions or deletions. So say you say that gorillas and chimps have a specific deletions that, or we have chimps, let's say, and humans have a specific deletion that gorillas don't have. You can see how there could be a derived character there, right? So the other reason why you want to actually find this is so that you can align the remaining part of the sequences so that their comparison is significant. So for example, look at these three sequences here. If you keep them as is, now here you say, oh, at this point everything is different. But now if you shift them, taking into account the deletion and insertion and so on, now you will see that now even this part are the same, even though they look very different, right? Because obviously the deletion here would cause a completely change, a shift in the framing here when it comes to comparison. Once it's done with that, then you will basically a program. This is all done by softwares, obviously. You don't do it by eye, but the program would take into account all of these deletions, all the nucleotide changes, and so on, to figure out a percentage of similarities between different, in this case, DNA sequences, which will reflect the relationship and eventually the evolutionary distance between the different organisms. Now, this data will be used using cladistic and then still the parsimony assumption. So basically, if you change, there is one deletion happening, for example, it would assume that that deletion, or trying to find an explanation and place all the species by assuming that that deletion had happened only once through evolution, not multiple times, which is parsimony, right? Then you could still figure out what came first or after in terms of any change, so what is ancestral and derived, by comparing to an outgroup, for example. And then there will be a maximum likelihood method that then ultimately would basically bring all these species in their appropriate place in a cladogram or a phylogenetic tree. Now here is an example instead using proteins. Now proteins have been used to further define the relationship between organisms, and you see that phylogenetic trees have changed since the introduction of molecular data, whereby some organisms that were put, let's say, unrelated to each other, now molecular data show that indeed they are part of the same family or same genus and so on, right? So this is, for example, cytochrome C. So this is from the mitochondrial respiratory chain, so it's fairly preserved, I would expect. But now you can compare actually organisms that are fairly distant to each other, like human, with yeast, so the plants, fungi, and so on. And so now these actually can easily be used to place all the organisms, so just based on the sequence of cytochrome C, and group them together and figure out their relationship. If you look, we don't have the time here, but you will see that this indeed reflects, look at the turkey and chicken, what was already, so duck, penguin, so these are all obviously birds. It reflects indeed the similarity like dog, seal, bat too, they're all evolutionary related here, reflects the classical phylogeny and adds some more information to that. Now, as I said before, this has helped also figuring out some conflicted hypotheses that were generated by the previous systematic. For example, here, by using these phytochrome genes like PHYA and PHYC, now these two species, Amborella trichopoda, and so that were actually first considered actually fairly separated, and then they were being finally placed as a group evolutionarily to actually where they belong, so as an early example of flowering plants or angiosperms. This is another phylogenetic tree that, for example, has been produced using ribosomal RNAs. As you can see, ribosomal RNA is indeed a type of biomolecule that is highly conserved. There are only a few of them, and they have an essential function, right? Ribosomes, they do translation, so they cannot be the product of excessive variability, and therefore there is very little variation, and therefore you can use RNA sequences only to study organisms that are very distantly related, such as in this case you're talking about organisms, and now they are the ones that basically you can study the relationship of organism to the original domain, so far away, the origin, very early splitting here in terms of evolution. You could not use a ribosomal RNA to separate a human Neanderthalensis from a Homo sapiens, something like Neanderthalensis from a Homo sapiens. We are too closely related. There will be basically no differences there, no meaningful differences there. And finally, last slide before the summary here. So phylogeny here, when it comes to molecular data now, then the phylogeny is done using the similarities between these different sequences, which then are interpreted through different statistical methods. We already mentioned parsimony, so assuming that any change should be explained with the least number of evolutionary events. Then there is the Bayesian probability, that basically what it says that the data should be interpreted using not only what you see in the data, but also adding prior information. Okay, so you should not just use the data, that's it. There should be also previous information based on other observations that should be entered ultimately into the final analysis. And then the bootstrap statistical analysis is basically a method in which data can be analyzed, you generate means, you generate multiple means. It's just a pure statistical thing. So there are different a lot of strong statistics behind the analysis of all this molecular data, things that now are integrated in software, which can be different actually also on the different type of methods that they use, statistical methods that we mentioned here. So in summary, what did we talk about in this lecture? Well, first of all, we talked about what systematic is and how it's in charge of classifying organisms and define their phylogeny, so their common relationship in the evolutionary tree. Then we talked about the classic classification, which is the lineage system, which has been around since the 1700s and is becoming a little bit outdated, as we mentioned when we were talking about cladistics. So we say how all the organisms are classified based on taxa, which correspond to hierarchical groups, which includes progressively less and less and less organisms, going all the way from domains ultimately to species. Then we talked about the characters they're used to do classification. So we talked about the characters, the homologous, analogous characters, and how they relate to different type of evolution. We talked about convergent, parallel, and divergent evolution. And then we define also what ancestral and derived characters are, and how can you figure out what is ancestral or what is derived when you look at a specific character. Then we talked about phylogenetic trees and how they represent not only the relationship between individuals from an evolutionary point of view, but also the time distance between them. We introduced also cladistics. And we talked about how cladistic is nothing else than a sub-branch, if you want, a systematic, which instead of using both evolution and morphology So derived characters ultimately and ancestral characters to group organism into taxa suggests that classification should just be based on clades, so monophyletic type of taxa. And so that generates particular types of phylogenetic trees referred to as cladograms. We'll go through that on a live meeting. We talked about clades, and also we talked about how phylocode corresponds to these new system that they are suggesting should replace the linear system of classification. And finally, we went through the molecular data. So what type of sequences can be used to further define the relationship between different organisms, going beyond just morphology, okay, going beyond the product of coding gene and phenotypes. So we talked about how DNA, RNA, protein sequences, so both mitochondrial and genomic DNA can be used. We talked about how each one of them, depending on how much they vary throughout evolution, can be used to establish close or distant relationship between organism, what are the pros and cons, and so on.